All right, for this video, we're going to work on genetics questions 23 through 37. The first set, 23 through 26, are made up. In other words, they're not real traits, which I try to avoid. But anyway, they can at least show you how certain crosses are done. So we're going to look at number 23 first. Number 23 says, in tribbles, crossing a white tribble with a black tribble results in all the offspring having black coats with white spots. First of all, you need to be able to identify that this kind of a problem where you're crossing two different phenotypes, white with black, and you end up with kind of a, instead of a blend this time, you can see both black and white in the offspring. This is called codominance. And you work the problem the same as you would for incomplete dominance. If this were incomplete dominance, you wouldn't see black with white spots, you'd see gray, because that's kind of in between white and black. So the first thing that you need to do for this question is to make a key. So if you're going to have a white rabbit, they're going to have two white alleles. And in order to have black, they're going to need two black alleles. The heterozygous condition, where you have one of each, is going to be spotted. And the question is, what will be the expected phenotype ratio when you cross a spotted tribble with a black tribble? So we're going to cross a black with a spotted, and I want the phenotype ratio, in other words, what the traits look like, black, white, or spotted. So whenever you've got a homozygous crossed with a heterozygous, your genotype ratio is going to be one of each. So half and half. Half will be big B, big B. Half will be big B, big W. The phenotype of this one is black. The phenotype of this one is spot. So you're going to have a 1-to-1 ratio of black to spot. The thing with codominant and incomplete dominance is that your genotype, which is the letters, that ratio, which here would be 1-to-1, is going to match your phenotype ratio. That's not the same if one of them was dominant. So for number 23, the answer is B, 1-to-1. For number 24, if we cross two spotted tribbles, so if we're going to cross two spotted, it's going to be BW crossed with BW. And I want to know what the genotype ratio is again. And again, you're going to see that the genotype ratio and the phenotype ratio match. So I think we're looking for the phenotype ratio. But we'll get both. So with this one, since you're crossing two heterozygous, you're going to end up with a 1-to-2-to-1 ratio, just like you would if it was a dominance question. And I can show you what that means with the Punnett square. You're going to end up with one of the homozygous B's, one of the homozygous W's, and two of the heterozygous condition. So that's what I mean by 1-to-2-to-1. Now, if this was a dominance problem, these three here would all exhibit the dominant trait. This one would be the recessive trait. But when you've got codominance, you've got a 1-to-2-to-1 ratio for the phenotype as well. So you've got one who's big B, which is black. You've got two who are big B, W, which is spotted. And you've got one who's WW, which is white. So you still got a 1-to-2-to-1 ratio, so the answer to number 24 is A. For 25, the trait of an extra long tail is dominant to a short tail. Two long-tailed tribbles are crossed. So I'm going to cross two long-tailed tribbles. Without any more information, since long is dominant to short, this is what I know of that genotype so far. And what we find out is that we get 352 long in the offspring and 118 short. So if you should be able to just glance at that and immediately see a 3-to-1 ratio, dominant to recessive. When you have a 3-to-1 ratio, you know that your parents were heterozygous. So that means these two parents are heterozygous for that trait. And then for 26, a spotted tribble with a long tail homozygous for long. So a spotted tribble, if it's spotted, it's going to be BW. And if it's long homozygous, it's going to be big L, big L. Is crossed with a black short tail. So black, here's my key, is going to be big B, big B. And then short tail, the only way it can have a short tail, which is the recessive trait, is to have two short alleles. What are the expected phenotypes? Without working a large Punnett square, without even working two Punnett squares, if you remember the ratios, this one goes pretty quick. You're going to have, you've got a heterozygous and a homozygous. Whenever you've got a heterozygous with a homozygous for color here, you're going to have one that's BW, which is spotted, for every one that is black. So that's half of them are going to be spotted, half of them are going to be black. And then for the tail length, all of the offspring, this parent's going to contribute a big L, this parent can only contribute a little l, so they're all going to be long. My question is, what are the expected phenotypes? Half are going to be spotted and long. The other half are going to be black and long. So what we're going to find is both spotted and long and black and long. The answer is C. All right, the next group of problems are sex-linked. Sex-linked can be a little bit tricky because you have to tie the trait to the X or Y chromosome. That's what makes them sex-linked or X-linked. I kind of use the X-linked a little bit more frequently because it's a little more descriptive. All right, so we're going to look at number 27. Two colorblind people all have colorblind kids. And it said on the page before that you should know that colorblindness and hemophilia are both sex-linked recessive. So when you're making your key, make your key for the trait that we're looking at, which is colorblindness. Big C is normal. Little c is colorblind. So colorblindness is recessive. And since it's also X-linked, we're going to tie this particular allele to the X chromosome because males only have one X and females have two, so we have to do it this way. So let me show you what the parent cross will look like. If we're crossing two colorblind people, so first let's start with the female. She's XX. And she's colorblind. If she's colorblind, she has to have two colorblind alleles, so two little c's. And then the father is also colorblind, so he has a colorblind allele on his X chromosome. There's nothing on the Y. So because these are X-linked, that means that the colorblind gene is linked to the X chromosome. It's found on the X chromosome. So if we look at that, and the question is, will two colorblind people have all colorblind kids? Well, the female can only contribute a colorblind allele. The male can only contribute a colorblind allele to the female or a Y to the male. Notice that all of these kids, both of them, are colorblind. So if two people are colorblind, all of their kids will be colorblind. 28 says, okay, this is kind of one where we've got several generations and a lot going on. So a woman has parents with normal color vision and a colorblind brother. So let's start with her. What we know about her so far is that she's XX because she's a female. Both her parents both had normal color vision. So let's look at her parents. Her mom and her dad both had normal color vision. So if dad's going to have normal color vision, that means his C is going to be a big C. If mom is going to have normal color vision, she's going to have a big C in something else. Now, the other thing that we know is that she has a brother who is colorblind. So let's do her brother kind of at the same generation level as her. If he is colorblind, he has a little c. So he got the Y from dad. So where did he get this little c? He had to get the little c from mom. So that means she is a carrier for colorblindness. Now, what do we know about the woman? She marries a colorblind male. So if he's colorblind, he has a little c. And they have a colorblind daughter. So they have a daughter who is colorblind. That means she has 2 colorblind alleles. She got one of those colorblind alleles from dad. She got the other colorblind allele from mom. Okay? And mom would have gotten this colorblind allele from her mom, meaning that she got the normal allele from dad. So there's everybody's genotypes. What's the probability that a son will also be colorblind? So let's say this same couple has a son now. He's going to be XY, and we're wondering what the probability is that he will be colorblind. All right, so if we make a Punnett square with these 2 people, and I always exaggerate the sizes of my C's so I don't get them mixed up, because C looks the same, big and small, if you're not careful. All right, so in here for the female, we're going to have a big C, little c. Here, we're going to have little c, little c. So I want to know the probability that a son, so we can eliminate the females from this part of the equation. What are the chances that if they have a son, the son will also be colorblind? So here's the son. There's a 50-50 chance that their son would be colorblind. If the question had read, what is the probability that they'll have a colorblind son, then it would be 1 in 4. So the answer that I'm looking for is 50%. All right, on to the next. So let's look at question number 30. Hemophilia is also a recessive X-linked trait. So capital H would be normal. Little h would mean hemophilia. Hold on 1 second. I gotta answer a text. Sorry about that. Okay, so 30. A woman who is a carrier, if she's a woman and is a carrier means that they don't have the condition, but they carry the allele that does, but it's masked. In other words, it means that she is heterozygous. So she carries the disease, but she doesn't have it herself. She marries a man with normal blood clotting with respect to hemophilia. There's all kinds of blood clotting conditions, but we'll say we're talking about hemophilia here. So he's normal. Can they have a son who's a hemophiliac? In other words, we're wanting to know, can they have a son who's a hemophiliac? Sons get their Y's from dad, so dad's going to give this son a Y. Can the mother give him an H, a little h? So yes, he can get hemophilia from his mom. X-linked traits are passed to sons from their mother because they get their X from the mother. All right, 31. A male hemophiliac, let's put him first. He's male and he has hemophilia, so we attach that H. Marries a woman who is homozygous dominant for blood clotting. So she is homozygous dominant for hemophilia. What type of children can they expect? All right, I'm going to show you the shortcut for making a Punnett square for this because I think it just is easier to see without all those X's. You can put the X's in if you like. I don't like. So I'm going to take the male, his gametes, he can give me an X with an H, a small h attached to it, or he can give me a Y. And I'm just using the Y as kind of a place marker inside my Punnett square. The female can give me an X with an H or an X with an H, as far as big H. Then I can look at them together. To me, this just is a little less busy, and it goes faster too than writing all those X's and Y's. So if you have two H's, you know that's a female because she has two X's. If you have an H and a Y, you know that's a male. So my possible offspring are two females who are carriers and two males who are normal. Carriers are normal as far as their phenotype. So to answer this question, all of their children will be normal if that's the parents. All right, I'm going to make another video for blood type so that this one isn't too busy.